त्यसपछि तपाईको मास्टर्स गरेपछि युकेबाट बायोमेट्रिक्समा मास्टर्स गरेपछि I went back to Rampur. Actually, I taught in a postgraduate program. They had just started a PG program, and actually I developed some of the courses there. So I was kind of felt lucky that I was involved in that program. So त्यसपछि मैले 98 I left IAS. Then I went to ISIMOD and CIMIT for 2 years. Then I came to States in 2002 for my PhD. I completed my PhD in 2006 in forest biometrics with minor in statistics. Then I started looking for jobs, and obviously health science was one of the areas where we had a lot of opportunities in statistics and data analysis. So that's how I ended up in health science field actually. And I had actually taken some clinical trials and survival data analysis type of techniques that would be more relevant in health sciences. So that's how I started working in University of Colorado, moved to University, St. Louis University, and I also worked at Harvard School of Public Health. Now I'm at Johns Hopkins University School of Nursing as a statistical consultant. I teach some courses and I do a lot of consulting. So in a way, I have left agricultural science or agricultural research for some time, but I kind of, in a way, in my heart, I have kind of my love with agricultural research in a way. So I have kind of forgotten a lot of agricultural stuff, but then I'm happy to be back in a way. So I'm gonna do some, go over some of the basic elements, or I guess the topic, it's very general. So it's not gonna be specific and it's not gonna be very detailed. And you have actually gone over some of the ideas of research design and statistics and survey research, those kind of ideas before. So I don't have to repeat those. And it doesn't hurt to reinforce some of the ideas, I guess, but then I would not consider myself an expert in survey design or social science research or something like that. But then some of you probably are doing some of that kind of research as well. That's what I would guess. Then I'm kind of more, I would say, biometrician in a way. I have seen a lot of biological science problems in a way. So, but then I lack a lot of basic understanding since it's been long that I have left agricultural science anyway. But let me try. I'll go through some of the ideas that I'm trying to present today. And you can just jump in and ask any questions at any time. Then if you have any questions, then you can send some questions in your chat box. Then some of the senior colleagues here, they might be able to help you. I might not be able to monitor all the questions and comments, but I'll at the same time try. Let's see how it goes. I have taught via Zoom, but it's not always friendly. So just stop me if you have any questions or comments. All right, so outline for today. So I have an hour or so, I guess, I would say less than an hour. So I'll try to be brief. So my plan is to do some general introduction, or I guess discussion. Then I would like to spend some time today talking about the data analysis plan. So this is something new in a way. So we always do talk about protocol, trial protocol, and proposals, that kind of things. We always specify what the design is, what the variables are, like what the instruments are, what the scales are, that kind of thing, right? But then having worked in health science research a lot, then I have come across a lot that we have, or we are supposed to do, or we are supposed to prepare our data analysis plan. So that's a key, that's a very important aspect of data analysis, rather than just collecting data and going straight to data analysis. So I'll briefly talk about that. And a part of data analysis plan would be data management plan and data analysis plan in a way. Then time permitting, I'll try to talk about briefly, talk about the interpretation of results. There is a lot of issues with presentation and I guess the interpretation of results from the statistical analysis. So it won't be comprehensive, but I'll try to bring you a couple of issues with that. All right, so you have already seen this before, research methods. So some of you actually have expertise in research methods and actually have specialized in research methodology. So I don't have to go over all the details there. But the part of research methods, it's a comprehensive, also a huge topic in a way. So statistics would be in a way part of research methods. That's how I see it. So then everything in research starts with research question, right? Research problem. If there is no research problem, then you would not be doing any research, or you would not have any research questions. So to collect your data or to answer your research questions, so you will have to have research design appropriate. So that could be experimental, could be non-experimental, it could be observational. So you are all familiar with that concept, but I just briefly wanted to talk about that. And very important component of research design would be measurement. In biological sciences, we don't talk too much about measurements, but in social sciences, then measurement is a very huge topic in sociology and psychology, right? Measurement is very, Dr. Bhandari and others actually know this better than I do, but this is a very important aspect of research methods. Then the data analysis. So when we say data analysis, it may not be all standard statistical methods. So there may be other techniques that would be considered part of data analysis in a way, but in a way, in this conversation, we'll use data analysis as like a synonymous to statistical analysis. Then interpretation of results and reporting of findings, how to report the findings. So I'll briefly talk about that as well. So actually, this is a schematic diagram of level of evidence. We don't see this much in agricultural research. This slide actually comes from an example from School of Public Health. But the idea here, I don't like everything here, but in a way, I just wanted to give you a brief idea. So it can start from animal research or even lab research. Can go all the way up to like randomized controlled trials, RCT, that's what they typically call. We call experiments, right? Experiments is like almost synonymous to research. That's how we are trained. But then there are a lot of research that could be qualitative or observational, but then at the top of the pyramid would be meta-analysis. So that would be the highest level of evidence you can get in terms of scientific evidence, right? In a lot of social sciences and public health and biomedical research, meta-analysis is very popular. I mean, I have seen in agricultural research as well, but then it's not like a combined analysis. So you have heard combined analysis over site and combined analysis over time, right? So in that case, what we do is we pull or we use raw data. We use study individual plot level data, or I guess the treatment level data. So if you do that, that's not meta-analysis. What meta-analysis is basically it's a study level data, then you have multiple studies, you combine study level data to come up with a meta-analytic estimate. So that's what meta-analysis is. So that is the top, right, in a way. So what we do is probably experiments or like observational studies. That would be somewhere in the middle, I guess, somewhere here. I mean, I don't like everything here. I mean, it says observational studies. Some of these animal research can be experiments too, right? But then in terms of schematic diagram, that's how they present it. So I just wanted to bring it up in a way as a part of, so if you move up in your pyramid, then that becomes a stronger evidence of scientific rigor in a way. All right, so we talked about research design, so I'm not going to repeat everything, but then we have in agriculture research experimental design. That is very popular, I guess. Then in other areas, then quasi-experiments and non-experimental research is also popular, right? Then you can have also quantitative versus qualitative, or even like a mixed methods type research that you have seen before from Dr. Bhandari. All right, so in terms of measurement, I just wanted to briefly, so if you read research methods book, then you'll kind of see this, right? In terms of accuracy and precision. Sometimes we confuse accuracy and precision, right? Precision is related to variability, right? So if you have high variability, you have low precision, right? but the accuracy is related to bias. So if you were to target, shoot a target, then if you are consistently shooting at a target, then you have high accuracy, right? So you may be shooting consistently close to the target. I mean, that's high precision and high accuracy, right? But then you may have good accuracy, I guess. I don't want to say high, but that's good accuracy. But then the, I mean, you are shooting around your center or the bullseye, I guess. But the shooting is a little bit spread around in a way, right? So in terms of precision, then that is low, right? So you don't want to be here in a way, right? So you want to be here in a way. So we don't know exactly where the target is in a way in terms of measurement, but we try to do our best in a way. So that's the idea in terms of measurement, all right? So you have gone through the principles of experimental designs, like why you would need replication, why you would need randomization, then why you would need blocking and mechanism for local control, that kind of thing, right? And these three would be probably very common. I guess RCBD would be the most common design, right? Randomized complete block design. That's very popular design in agricultural experiment. I mean, that's what I used to, or we used to do at Lumley. So another aspect of data analysis or part of statistical study or statistical design would be sample size. In experiments, we talk about like error degrees of freedom, right? You don't want to have very small error degrees of freedom. You want ideally, let's say 10 or more than 10, 12 or more than 12. So that's, I guess, a rough guideline for how much replication you would need given certain number of treatments, that kind of idea, right? But then if you are doing a survey or other type of studies like RCD in social sciences or epidemiological studies in veterinary study, animal studies or something like that, then you would actually need to calculate sample size, right? So for that purpose, for sample size estimation, then you would need three things, right? First thing would be power. So you would need at least 80% power, right? You would need more than that, ideally, like 90% or so. Level of significance, we have learned it's commonly 5%. Then what is important is the third, that we are not always we don't always talk about. What we need to talk about is the effect size of importance. So what we are doing is we run experiment, collect data, then we perform some statistical analysis, and we say statistically significant or statistically non-significant. And we don't really talk about the effect size, although we talk about like doing confidence intervals and that kind of thing, right? So statistical significance depends on several factors, and one direct factor would be sample size, right? If you keep increasing your sample size, even a tiny bit, a very small effect size is going to be statistically significant, right? So then as a researcher, you'll have to think about effect size. What is the minimum difference between two varieties in terms of yield that is going to be meaningful for recommendation, right? So is it like 10 kg kilogram per hectare? Is that a meaningful change or the difference, right? May not be so, right? So you'll have to think about that. I mean, if you keep increasing your number of replications, then even 5 kilogram per hectare difference between two varieties can be statistically significant, right? So the point is, what is the minimum difference that is going to be meaningful to you as a researcher? So if you were to recommend your new technology to farmers adoption, then what is the minimum difference? What is the minimum effect that you would consider to be important? That's effect size. That has to be really thought through as a researcher, right? So that can be difference in terms of means, or that can be difference in terms of proportion. Sometimes we analyze as a ratio of proportion, like a relative risk, those kind of things, depending on design and the problem and the nature of the data. All right. So difference between two means can be raw or it can be standardized. And one of the popular standardized measures would be cohesity. That is kind of very popular in social science areas. But we can calculate in, I guess, biological science as well. But what is important is the confidence interval. So what point here is to estimate sample size, you would need three things, right? You specify your effect size that you would consider to be important, then power and the level of significance. So given those three things, you can estimate sample size. Then you have many programs and packages that you can use. So in terms of statistical analysis plan, then let's say you collected data, you had a very rigorous method of data collection instrument or way of collecting data. Then how do you analyze? What I have seen, or what I even tend to do is just run a test, right? So that's not a very good idea, right? So first you'll have to do the data quality checking and derive variables and do all sorts of exploratory analysis in a way, like initial analysis, I would say like a preliminary analysis, right? So you'll have to do that. So part of that would be a good practice in statistical analysis, right? So that is driven by your research question and type of hypothesis and the design, right? Your design will affect your statistical method and your analysis plan. I guess what I'm saying is your analysis plan has to address these I guess bullet points or key areas of topics basically. And then you'll have to describe your dependent variables and independent variables and the covariates, like some sort of confounders or some other ancillary variables, right? Let's say you ran an experiment in a field and cow grazed over a little bit, or there was some flooding. So you'll have to collect that data as well in a way that you can use later. Then the part of this would be data analytic method, which is what we're going to emphasize today in a way. And you want to start always with a simple method, exploratory or like a univariate method, then move to like more complex method, like a bivariate or like a multivariate method. And part of analysis plan is to do what type of a table you're going to produce. I mean, not all tables will go to your manuscript, right? So all the analysis table are not going to a manuscript, right? But as a part of your analysis, you'll have to produce a series of table shells even before. I mean, you'll have to think through what kind of tables, what kind of analysis I'm going to run, then fill these tables in a way with the summary data or the analysis output in a way. So that's very common to produce table cells, and you fill that in later after analysis. Then part of analysis plan would be specifying what statistical programs you plan to use, right? I mean, you can always change later that we use a different program. Just be honest what you are going to do, right? Or what you did. Basically, popular statistical programs you have heard from others as well would be SAS, SPSS, and like S+ or R, right? There are other experts who know this better, but personally, I use SAS and SPSS. That's what I do. But then I had been using other programs as well. Yes.
Mute गरिदिनुस् त please, Susma बहिनी.
Thank you. All right. So if you need help with other programs like R, and there are other experts, probably Dr. Budhathoki would be a better resource person for R, yes, but I've been using SAS and SPSS a lot. All right. So in terms of data management, your plan should address some of these points, like data quality checking. So once you have data, then instead of directly running a t-test or analysis of variance or regression, just plot your data, right? Look at the distribution. That could be in terms of histogram, or that could be a scatter plot. Look at the shape and look at the variability if there is any outlier. That kind of analysis would be always very helpful. And you can always do descriptive statistics, right? So start with calculating like mean, median. I mean, you would think that that would be a very naive thing to do, but start with that, and that always helps, right? Rather than just jumping into like a modeling, like running a very complex multivariate model. And always look at the missing data. It's inevitable that we'll find some missing data in our study. Always, like respondents refuse to answer some of the questions, or you lose some data during data collection, right? And always look for two things: pattern and extent of missing data. How much missing data do you have? Is it like 5% or less or more than that, right? Then if you have more than, like if you have substantial missing data, then is it a good idea to impute missing value estimation and plugging that into your missing cells in a way, right? And you can do a lot of things with the pattern, missing data pattern, but let's not worry about that today. Then the coding of missing values. Sometimes in survey research or in other types of research as well, we specify certain values as missing value. Like if you have a missing value, then they would code like a -99, right? So that's a good practice. But then if you don't exactly know how to handle that during analysis, then that could be problematic, right? So in your analysis program, you'll have to specify that is my missing value. If you don't specify that, then that could be even worse. Actually, that missing value code actually goes into your analysis. So if you don't really know how to do the missing value code and the analysis of that, then it's better to leave it just blank. And most of the programs, statistical programs, we handle or we treat that as missing data, right? Then if you're going to impute missing data, then that's a different technique, all right? So then you'll have to always calculate other variables. That always happens, right? You collect your data, yield data, or biomass on a per plot basis, then you want to convert that to a larger acreage, like a per hectare basis. So that's always calculated, and just make sure that is correct. Like in survey research, if you have many items, then you would calculate scales and subscales. All right. Then another good practice, if you are managing a lot of data, then the version control. That's very important, right? I mean, at least you have like a raw data and one or two maybe analysis data sets, right? But then if you are dealing with a very complex data structure or many data points, then version control is very important. Even the output, or I guess the programming code, those kind of things. And even after producing some output, data analysis output files, it's a good idea to have some mechanism with the version control, right? And personally, what I do myself is I date stamp everything, right? I date stamp so that I would know when that was saved or when that was created, like the output file and the program file, those kind of things, right? I mean, it's a matter of like working what works best for you in a way. It's not that you have to do it, but I guess the general advice would be you have to have some mechanism for version control, and you have to document that. If you ever need to go back and look what you did like 2 months ago or like 2 years ago, then you can always look at that document, some sort of like a paper trail in a way, right? Then you have to have a good file management system, right? Where you store, is it like a server or your local drive, that kind of thing. It's always important. You think like a statistical analysis is like a very brief exercise. Then to get to that point, to prepare your data set, you have to go through, you have to follow some very important steps that would be considered like a good practice. And in complex data centers, what they typically do is they even like give you some guidelines how to name your files and how to name your variables, right? Maybe for mini grant, that is too much. But then as a good practice, then just think about how to systematically name your files and how to systematically name your variables from your research in a way. Just think about that. And codebook, prepare a codebook, what the name of the variables are, what the levels are, right? How you code it. Did you code 0 and 1, or did you code 1 and 2 for male and female? So just document that, right? And that always helps. And if you have very complex data structure, then you can also have metadata. Metadata basically means data about data, right? So if you have just a few files, then that's okay. But if you have many files, then it's a good idea to have data about your data as well. That's metadata. So you got your raw data set, original data set. You want to do some internal checking or quality checking, and If there is a typo, you want to always correct that. Once you do that, then that is your raw data set, right? So then copy that over to another format, format meaning another file, and manage that and prepare analysis data set, right? You can have intermediate and other types of analysis data set, but then idea would be never use your original file as an analysis data set. That's always a good practice, right? Always leave that alone in a way. That's your original data set. Then create your own data set for analysis. That's analysis data set, right? That's a good practice. So as a student of agricultural research, or I guess, or a statistical analyst, that always would be a good practice in a way, right? So then thought would be then if you prepare your analysis data set and you have your code book or the documentation, then, and if you hand over that data set to somebody else, then would they be able to replicate what you found using the same method? So that would be the key idea, right? And in data management centers, this happens a lot actually. So you'll have to replicate things. Okay, so in terms of analytic methods, then consider some of these key points, I guess. Like the terminology may vary, like if you are from agricultural extension or agricultural economics, statistical terminologies may be different from other areas of biological sciences, right? So as long as you follow the guidelines or good practices, it's fine. Like in economics, or I guess social sciences, they would typically call like a panel data, but then our public health, other epidemiological studies would say longitudinal data or longitudinal study, that kind of thing. So it's a matter of terminology in a way, but terminology can be slightly different. Like mixed modeling and multi-level modeling ideas are very similar, but then terminologies are slightly different. The point here is depending on your area of research, or I guess area of concentration, your terminology for research, or I guess the statistical method, can be slightly different. What I'm saying is you don't have to use the same terminology across different fields, basically. Then there is a plethora of standard statistical methods that you can choose from in a way, right? Then there are some alternative methods, or there are some competing methods that you could be equally valuable in a way, right? But then certainly in some cases, then some methods are better than others. One key point I wanted to make is it's not, I guess, very popular in agricultural research is non-inferiority testing or equivalence testing. What we do all the time is what is called superiority testing. So we want to always show variety A is better than variety B, or treatment A is inferior to treatment B, or something like that. We always want to see better or worse, superior or inferior. What can happen, or what is possible, what is also possible is you can test for non-inferiority or equivalence. It may come in some cases where, let's say your standard variety is very well adopted, very good, then you want to develop a new variety that is as good as or equivalent to the standard, but it is better in other aspects. Maybe it's grain quality, or it's cost of production, whatever, that could be beneficial, right? So if you want to do that, then you can still keep your yield as a crop yield, or grain yield as a like a primary outcome variable. Then you can use other as a like a justification for what happens. What I have seen in my areas these days as well, people claim, or people say two varieties are similar based on statistical non-significance, right? So statistical, if two varieties are statistically, they are not different, then they would conclude that they are very similar. So in a way, technically, that's not an evidence of equivalence. That's not an evidence of non-inferiority. So you'll have to actually follow different design. I just wanted to bring this idea, basically. So my point is what we have been doing a lot is superiority testing, right? So then a change in statistical methods also can change inference or the conclusion, right? So like if some analysis is more appropriate for analysis of covariance, then analysis of variance would not be a better method in a way. Or what I'm trying to say is analysis of variance and analysis of covariance will give you different answers, basically, right? Choice of statistical method basically will affect your inference, right? And as you know, research design flaws cannot be fixed by any advanced statistical method. So data analysis is limited by the design, right? So if you made a mistake with the study design, then you cannot fix that with the analytic. And in some cases, you can improve your analysis. Like if your blocking was not very good, like blocking was imperfect, then there may be some covariates that you can actually collect. You can have some covariates and use that as like a covariates in your adjusted analysis. You can do that, but then it cannot actually fix that you missed will design experiment that you would have done or you should have done with the blocking, right? So design is always very important. So then further considerations. So in terms of analysis, you'll have to consider some of those things that I have already talked about. Then another would be the nature of the variables, right? So is your dependent variable continuous? Is it approximately normally distributed, or is it categorical variable, like a nominal or ordinal type variable? Or your outcome is actually time to event, time to until something happens, right? Time to somebody dies, right? Time to some event in life course. So if you are analyzing that type of variable, then your analytic method would be very different. So your data analytic method is driven by the nature of your dependent variable as well as your independent variable. But in terms of independent variable, it's much more flexible. What drives is a dependent variable or outcome variable, right? So you have learn descriptive versus inferential statistics. I would always start with descriptive statistics, right? Typically with statistical programs, if you run inferential statistics at the same time, you can ask for descriptive statistics, right? From that point of view, you could actually directly go to inferential statistical methods, but then that's not a good idea, right? Part of your analysis variance analysis, you would get means and standard deviation. But then I would always start with calculating means and standard deviation, then move to analysis of variance. That's what I would do. I would do correlation bivariate analysis, then I would move to multivariate analysis. So the point is, don't just jump to like more complex analysis. Then if there is a designed treatment structure, then I would use planned comparisons rather than like doing multiple testing, like all possible pairwise comparisons. So that's not a good practice, right? So if there is a good treatment structure, like you can do all new treatments versus your control or comparison group. Then if that is significant, then you would actually do the comparison within new treatments, like new treatment A versus new treatment B, that kind of like contrasting your groups. So you could do that in a way. There are some statistical theories why that is not a good practice in a way, but as a recommendation, that's what would be a good practice. All right. So as I said, descriptive statistics, I would consider that as a part of your initial or exploratory analysis. Then part of that could be graphical displays, right? Graphical displays could be very simple, like as I said before, scatter plot, histogram, and line plot, those kind of things. Always start with that. That will help you a lot. Then if you are using parametric tests, then always check for assumptions. If you are going to analyze your data using analysis of variance or linear regression, always look at the histogram, right? Whether your data is approximately normally distributed or not. That's always a good practice, right? Like checking for homogeneity of variances. There are some tests that you could run for homogeneity of variance test. So it's a good idea. Then if the assumptions are violated, then you would not use a t-test or analysis of variance, right? You would go for other alternative methods. Like one way would be to look at the data transformation, right? You can take a log or a square root or whatever works for your data in a way, depending on the variable that you have. Hope is once you do the data transformation, the transformed scale would be approximately normally distributed. So then you can apply standard statistical methods like ANOVA or t-test, right? So if that fails, or some people don't prefer doing data transformation, right? Because when you report at the end, then you'll have to back transform. A good practice would be if you decide to do data transformation, report as both a transform scale and the back transform data. That's a good practice. Some people don't like that. Then you could use non-parametric methods, right? That do not assume normality. Like a lot of categorical data and ordinal, like a rank data, even scores and continuous variables, if they are not following normality or normal distribution, then it's a good idea to use non-parametric methods, all right? So you can use, or we commonly use p-values and confidence intervals. I encourage you always use confidence intervals. P-value is very limited. It's very common to use p-value, but then don't rely on p-values only. And statistical significance is not the same as practical significance. So I briefly talked about that, right? Your test may be statistically significant, but when you look at the difference, what the difference was significant in your analysis, that may be very small. Or sometimes even if your difference was very large, that you would consider to be very meaningful, then your test says that is not significant. That may be because of so many reasons. I'll talk about that later, I guess, if I have time. Then avoid data tracing. What is data tracing? Is you would continuously apply or analyzing your data until you find significant difference in a way, right? That's what like a data tracing is basically. Like you can try different ways to look at your data. Sometimes you dichotomize, or sometimes you trim your data. Sometimes you rank your data. You know, it could be a variety of ways that you can look at your data until you find statistically significant result. So that's not always a good practice. Why is that? You have to have a good plan, and you have to have good data collection practice. Then you specify your method a priori beforehand, then apply that, and it is what it is. So you'll find out what that is basically. So if that is not significant, just report that and try to interpret why that would be the case. All right, so in terms of exploratory data analysis, so I have a couple of graphs that I wanted to briefly talk about. So you would start with like a histogram, then histograms show something like this, then it's a good idea not to report, or you can report mean and standard deviation, but what is more important is it is better to report median and interquartile range, like a first quartile and third quartile as a measure of variability, right? Interquartile range as a measure of your variability. If your data is skewed, like a right skewed or left skewed, or positively skewed or negatively skewed, right? And your mean is going to be always toward the long tail, right? That's how it works in a way. So in this case, then you would not typically use t-test or analysis of variance, right? I mean, you can try transforming this, or you can use other alternative methods. All right. So what is important also is to look at another, that was like a smooth curve, right, from your histogram. And what you have here is basically here, another histogram, but in terms of bar, right? So we could superimpose our line there, but then there are not, like, your intervals are wider, so you can narrow your intervals and try to fit a curve in a way. So it looks a little bit skewed in a way, right? It's a like weight distribution basically. But if you have categorical data, then what you have is not histogram, that is bar graph, right? So if you have like a marital status and you are counting people, then that looks like histogram, but that's bar chart basically. And bars do not touch basically, right, in bar chart, but in histogram, bars touch each other. You can choose your intervals. You can define wider or narrower intervals, depending on how you want to present or how you want to look at. So these would be part of exploratory data analysis, basically. Sometimes in terms of presentation, bar chart, maybe what goes into your report. And you can also present box plots, right? Something like this. This is a single site, let's say. There were 2 sites. Don't worry about the cortisol. I guess it's a different measure. It's a stress measuring hormone, or I guess the chemical. But anyway, so point here is this is minimum. In terms of box plot, this is 1 box plot, right? And there were like 4 or 5 groups. And for each group, they have presented box plot. So what you have here is minimum, then you have maximum, then lower quartile and upper quartile, right? Then in the middle, you have median, right? So that's how you would present your box plot. Okay, so I'm not going to spend a lot of time talking about the analytic tools here, but the point here is what I'm classifying these methods as ANOVA type analysis. So if you have analysis of variance or analysis of covariance or t-test, that would be all ANOVA type analysis. What we are doing is we are comparing means across groups, right? Then your groups can be related groups, or your groups can be independent groups, right? If you are comparing males and females, completely independent. If you are doing before and after on the same subjects, that's related groups. Same groups, but different times, right? So there are different ANOVA models. I'm not going to spend a lot of time here. Basically, you can do, so factor is basically number of independent variable, right? 1 independent variable means 1 factor. 1 factor, 2 factor could be variety and fertilizer. So advantage of 2-factor analysis would be you could look at interaction between variety and fertilizer. Then in analysis of covariance, then you can look at 3rd variable. What we typically call is covariate, right? So like flood damage. So you ran a field trial, then there was some flood damage. Then if you have some data on flood damage, then if you can quantify, reasonably quantify flood damage, then you could probably use flood damage score as a covariate. I mean, this is just an example, right? And in terms of repeated measure or longitudinal measurement over time, if you are doing on the same subject, same animal over time, then over time observations are not independent, right? So you'll have to use repeated measures ANOVA or other like time-dependent methods. Okay. So again, parametric, under parametric methods, you have other regression type analysis methods. I'm just saying ANOVA type and regression type, right? These are very related methods, but then you would typically see them as like a separate methods. And correlation and regression, they are closely related, right? Correlation basically is an association, measure of association. With regression, you can make a prediction, right? And linear, when you have linear, then that also has normality assumption, right? So that could be simple linear, or that could be multiple linear. And for that, then what you would do is overall, you would look at the model, if model is significant or not. Then what is the direction? Is it positive, negative, or what is the size of the R squared, that kind of thing. I mean, there is a lot. What could I do in an hour, I guess? Then these are all called general linear models. So both ANOVA type and linear regression type analysis, they can be called general linear models. So a lot of, or I guess, if not all, analysis of variance type models can be analyzed using regression techniques. They are related, basically. Statistically, they are not any different, right? So in terms of non-parametric methods, I think Dr. Pokhrel in April presented this, so you are familiar with that. So there are a series of parametric methods, and if assumptions are violated, then you could possibly use non-parametric methods, right? And these would be non-parametric alternative methods. If assumptions are violated, if they are not normally distributed or So let's say homogeneity of variance assumption is not satisfied, then you could simply use non-parametric methods. They can be ranked data, or even if you have like a score or continuous data, then these techniques would rank your observations and work with the ranks. That's what it is. Like you may have heard Pearson's correlation coefficient. If X and Y are not normally distributed, then you can do rank correlation, Superman's rank correlation, right? Non-parametric methods, sometimes they help with small sample. But then problem is, not problem, I guess. My suggestion would be, if you can satisfy normality assumption, use parametric methods, right? If you can satisfy, because they are simpler and you can report mean and standard deviation, and they are more powerful if you can satisfy assumptions of parametric methods, right? But if you decide to use non-parametric methods, then you would report median and interquartile range, right? So you don't want to report mean and standard deviation when you use non-parametric methods. You always report median and interquartile range. What happens when you have categorical dependent variable? Your outcome is categorical. What we talked about before is basically continuous type outcome or dependent variable. If you have categorical dependent variable, then you can use something like chi-square test or Fisher's exact test, those kind of things, right? If you have categorical independent variable. But then if you have bunch of continuous or categorical independent variables, then you'll have to think about doing like some modeling, right? Like a logistic modeling. So if you have binary data, success, failure, pass, fail, right? Disease, no disease, or something like that, then you would do logistic regression, right? And typically you would report odds ratio. It's very popular in social science area. If you have count data as a dependent variable, you could also use Poisson regression modeling. Although we have not heard this very commonly in preliminary, like statistics courses. And these would be called like what is called family of generalized linear models. Basically, if you have count or categorical outcome variable, then you'll have to use what is generally called generalized linear model. Okay, so I'm not going to spend a lot of time talking about this. So you have learned this in basic statistics course anyway. But what is important is always think about the error, right? How precise is your error estimation, right? So if your error degrees of freedom is very small, like a 6 or, you know, around that, then that's not very precise estimate of your error, experimental error, right? So that has to be estimated with a reasonably larger degrees of freedom. So that's the idea. And depending on your structure of your treatment, then your models will be different, right? And this is just an example. And with the two factors, advantage of that would be you could look at interaction, right? That's the advantage. Rather than doing two different experiments with treatment A and treatment B, you could do that, right? You could do one experiment with treatment A and one experiment with treatment B. Then what you cannot study with that separate experiments is the interaction. So when you put them together in one experiment with factorial structure, is you can look at interaction. So I'm just quickly go over some of these, I guess, but I don't have enough time, but I'll try to move faster. What is interaction? So here, so if you have two factors, A and B, with two levels, A1 and A2 and B1 and B2. So what you have, something like a fertilizer and variety, let's say. So factor B is variety and factor A is fertilizer, let's say. Low dose and high dose or something like that. And variety A and variety B, let's say. So the difference between two varieties remains the same, more or less the same across two levels of fertilizer dose. Then there is no interaction, right? That's the idea. So you have learned that. So it's very, very important to understand and report accordingly in your findings. If you have a factorial structure, you can potentially have something like this, right? There is a difference between two varieties, B2 and B1 at two different levels of fertilizer. Then the magnitude is different, right? There is interaction in terms of magnitude, right? But sometimes what can happen is they can be different in terms of their direction, right? The difference actually gets switched. So that can also happen, right? Probably this does not happen quite frequently, I guess, but it's possible. So with blocks, you can have blocked experiment. This is typical RCBD, so I'm not gonna spend more time. So with split plot, you have two different error level, right? So for main plot, you have error 1, then for subplot and the interaction between A and B, you have like another error level. This is important to recognize, I guess. And the subplot level is more precise, right? Because it has more degrees of freedom. So table cell. So this is how you would design your cells before doing analysis, right? So if you have an experiment or survey, think about what type of table you want to produce, right? So rather than just running analysis and thinking about table later, think about, if you have not done analysis yet, think about the table that you want to produce, right? Not necessarily for your manuscript, but in terms of interpretation and I guess for your internal report. What are some of the tables that you want to fill in? So these would be table cells. And if you have experiment like a variety A, B, and C, and if you have a continuous variable like a plant height, then you want to typically do mean plus minus standard deviation, right? Or sometimes if that is skewed, then you want to report median and interquartile range, that kind of thing, right? If you have some categorical variable, then you want to do the frequency and percentages. That's how you want to present your categorical data. I mean, this is just an example, probably. So if you ran a rice variety experiment in farmers field and you ask what the grain quality was to farmers later, and they would rank or they would give you the response, excellent, very good, or something like that, then that's not a continuous scale, right? So you'll have to do the frequency and percent. And what is that is distributed within each column, basically within each treatment. That's how you want to, and you can do something like a chi-square or other tests for that. And if you have survey, then I mean, we have survey experts here, but typically we want to, table 1 typically would be like your participant characteristics, right? What is the sample characteristics? That's how we want to present our data, like what is the respondents distribution in terms of their gender or the age distribution. Typically you would do median and interquartile range. Age probably would be a little bit skewed, I guess. That's how they would report median and interquartile range. Like income is also skewed in a way. And if you have other categorical variables, then you would present like frequencies and percentages. Okay, so do I have a couple of minutes, I guess? So I ran over a little bit because I got started a little bit late anyway. So let me, I have just a couple of slides. So I'll go back and talk about interpretation of results. There are a couple of points that I want to highlight. So don't rely simply on p-values, right? P-value is just, it just says whether there is statistical significance or not, right? In terms of hypothesis testing. So that does not give you enough information. So what is more important is to look at the confidence intervals and effect size measures. So it is more important to look at the effect size measures, right? As I said before, your statistical test can be significant for so many reasons, or it can be non-significant for so many other reasons, right? And if you have a factorial structure, always carefully look at main effects and interaction. And I would start with interaction, right? Don't start with main effect. Look at the interaction. If interaction is significant, often main effects are not very important. If interaction is significant, just start with interaction. So if you have statistically significant result, then probably your life is easy, straightforward, and you are happy, I guess. So as a researcher, you want to see statistically significant difference, right? You are happy, you are going to report, I found statistically significant difference. And your journals, your target journal probably will accept your manuscript as well. But then remember, statistical significance is not the same as practical significance. Always keep that in mind. What happens when you have non-significant result? How do you interpret that result? Just say there is no statistical significance. And many researchers conclude that treatments are the same. So that should not be your conclusion, right? Your conclusion is lack of evidence of treatment difference, right? You don't have sufficient evidence to declare, to comment, or to say that there is treatment difference. You cannot really say that, right? But you cannot say treatments are the same using non-significant result, right? That's very important. You cannot prove null hypothesis based on non-significant test. And in terms of reporting, journal, follow journal guidelines, right? Like our NAPA journal. So we have our guidelines. So we would ask our authors to follow that guideline. So then as a NAPA mini grant recipient, then you would follow that guideline. So it's kind of specific to journal and organization. That's my advice. So rather than me telling you report in this format, I mean, there are some standard format like APA style and other styles, right? But then some specific journals may have their own guidelines. So then briefly talk about this, possible reasons for non-significant results, right? What would be some of the reasons that you are observing non-significant result? One potential reason could be measurement error, right? Your measurement was not very precise, right? Or there was some bias, or I guess there is no meaningful or absence of meaningful effect. There is some difference, but that's not meaningful. Intervention versus comparison, like a new treatment versus old treatment or something like that, right? Or there is a smaller effect than you had expected as a researcher or you had planned for. It's a possible reason, right? That could happen. As a researcher, you are kind of ambitious that you are going to see a larger effect, larger difference, but then in your actual experiment study, you saw smaller. That's why you your test was not significant. Or you used inappropriate statistical method, and that could be a part of your analytic technique that you want to review if there is anything that you could have done better in terms of using analytic method. There could be some design issue, right? Or there could be other confounding variables, like your randomization was not effective or blocking wasn't effective or something like that. It could happen, right? Or your sample size was too small, right? If your sample size is small, then you have low statistical power. So that could be one of the reasons why you saw non-significant results. And there could be other reasons, I guess. So if you have non-significant result, don't stop there. Try to find out if there is a reason behind it. Try to find that out, or at least bring that up in your discussion section, right? Like you could not recruit enough participants, or there were some issues with measurement or something like that. That could be. And I guess I would argue that you should be able to publish even non-significant findings, as long as you are able to interpret what they are or have a good discussion. So I think that's what I have. And thank you for listening. And I guess I admit that it was not very comprehensive or detailed enough, I guess, but it gave you probably an overview as a beginner. I mean, obviously we have very experienced researchers here, but I guess the target of this presentation was mini grant recipients, and hopefully you are able to take home some, I guess, suggestions or advice. And if you have any questions, you can always reach out to me at my Gmail. All right, which is here.
All right, thank you.
Thank you.
Yeah, thank you, Dr. Budhathoki. Thank you for your excellent presentation. I think this is helpful for all of us, not only research mini grant recipients, for all of us who are in here. And there are a couple of questions in the chat box. If you can address them, that would be great. Can you read them, or can you see them? Or
let me read. I have not looked at the chat box. Okay, let's start at the top. Okay, so Kaflezi is from Paklihawa, I guess. So thank you, Kaflezi. Okay.
So the first question is, could you please explain about probit analysis If you can be brief.
Right. So it's been a while that I have not done Provit analysis, but in entomological studies, they do a lot of Provit analysis, which is very similar to logit analysis. It's a matter of like using a different link function. So, right, instead of using logit link, you could use Provit link. And basically, yeah, it's a count data basically, right? You can use Provit analysis and Provit link to analyze your count data as well. I think it's very popular in entomological experiments, I believe. Maybe Dr. Parazoli would be able to contribute more on this. And I think Amrit Sharma.
Yeah, there is another question. Yeah.
So greater level of confidence. So if you were to choose confidence level, we would typically choose 95% level of confidence. Then you could also choose 99 if you need like a higher level of confidence or low level of significance, right? So you could do that, but it's common to use 95%. Sometimes if more error is allowed, you could also use 90% confidence level as well, but it's common to use 95%.
And there is another question from Amrit again.
Okay. So yeah, so in a way, as a beginner, you'll have to review your methods, your design. Like if you have CRD, then basically you have two sources of error, right? So if you have just group and within group, between group and within group, or basically treatment and within treatment. So with 42 treatments. So if you have 42 treatments, then 4 replications. I would guess you have a very large treatment structure. I'm not sure which treatment it is, but 42 sounds like, are you screening some chemicals or something like that? Yeah, 42 treatments. That sounds just huge to me. I don't know what, is this field experiment, or can you clarify this, like a 42 treatments?
Amrit, can you unmute yourself and yeah, briefly, don't
talk about that.
Don't elaborate much, but yeah.
Yeah, 42 treatments. Do you have 42 treatments?
So it's a two-factor research, and
okay.
we're using. Am I audible?
Yeah.
We can hear you.
We are using this botanicals and biocontrol combinations. So
I see.
In two different media. So it's like 2 growth media and 21 different combinations of treatment, and it becomes 42 in total.
21 times 2. 21 what? Sorry.
One factor is the combinations of different botanicals and biocontrol treatments, and the next is the growth media. We are using vermi and vermicompost and FIM in combination. So
right. So I would use just 2-factor analysis of variance, I guess. Then in terms of comparisons, then I would guess you'll have to do multiple comparisons for this, because you have like a 21 and 2. I mean, for 2, this is simply if that is significant. I mean, I would look at first the interaction. If interaction is significant, then I would plot your data. Maybe on horizontal axis, I would put like a 21. Yeah, something like that, I guess. Just look at the interaction first. And if that is significant, then you don't have to worry too much about the main effects. If interaction is not significant, then you'll have to interpret like a 21 means and 2 means separately for each factor, basically. Yeah, basically you are doing a F test, right? Basically, you are doing 2-factor ANOVA. As long as your dependent variable is continuous. What is your dependent variable? Outcome variable. You are on mute, sorry. Amritji,
No.
you are on mute.
I have some audio problems. So
Yeah. What
I
is your
cannot
dependent variable?
Hello?
Yes, go ahead.
We could hear you. Yeah, what's your dependent variable? Dr. Budhathoki is asking.
Can you, sir, please write it in the chat box so that I can reply?
Sure. Yeah. Let's.
Yeah, what I'm saying is it will depend on your outcome variable, right? So if you are, if you have continuous dependent variable, and let's say that is approximately normally distributed, then you can use ANOVA. Then if you are counting and if you are categorizing your responses, then you'll have to use different techniques. So probably you'll have to work closely with your advisor, or I guess, yeah, in future to figure out what model or what analytic technique would be better. So first you'll have to figure out your dependent variable and the nature and the distribution of your dependent variable. That will probably help.
All right, sure. Dr. Pura, I think there are a couple of other questions in the chat box. We know we have gone over time, but there is one question from Dr. Kafle. It is about, I think you can read it. Review of the published work is becoming popular. Is there any tips on how to pursue it? Its advantage and disadvantages. Before you answer this, Dr. Kafle, we already had one orientation on, or rather say, workshop on review of the literature. And if we have recording, we will be happy to share that with you. Dr. Wagle, I think you have information already, am I right?
Thank you, Dr. Ramji, for that valuable information. Like, one of the other things that I wanted to ask was that, is there a particular book written by one of us who know more about the agricultural research in Nepal, so that could be used as a reference material for academic purpose?
Excellent.
That was another question that I had set personally for Chakra.
I don't know a good resource for agricultural research systematic reviews. I guess I have other references that focus on like a health science, and they do a lot of like systematic reviews and meta-analysis. I can send you some references, but I'm not sure if I have any on agricultural research. So I'll try to find it out.
All right. So,
thank you.
I also have made a note, Dr. Budhathoki, if you have any kind of not only literature, if you have any publications or any other reading material that our students, research mini grantees can refer to in the future related to the presentation you just made, that would be great. It would be much appreciated. Yeah.
So I can post some references. And I can send you the slides that I just used.
Yeah.
And I think it's being recorded, so they can also watch this. I mean, this is not very comprehensive, so I should admit that upfront. I mean, upfront meaning almost at the end.
And there is another question from Pritam Thapa. Can you elaborate more of test of equivalency, comparing with superiority tests with example, please, and also comparison of looking in p-value and confidence interval.
Right. So what happens with the non-inferiority or equivalence test is you would specify the difference that you would consider the margin of difference that you consider to be negligible. Then you would use that as like a equivalence margin, and you would design your study, and based on that, then you would run some confidence intervals. So that's how you would do. But what we commonly do in agricultural research is we test superiority of one treatment over the other. But if you really want to do the non-inferiority, then you'll have to design your study slightly differently. You cannot just use ANOVA or regression to prove non-inferiority. So you'll have to use a different method. And that is typically done using confidence interval. And there are other ways to look at that, like a one-sided versus two-sided confidence intervals and other techniques that are kind of new in a way in statistical field as well, or I guess in RCTs, or I guess in health sciences.
All right, excellent. Are there any other questions other than posted in the chat box? If we can take one question, then we can close today's orientation.
Oh, I think Amritji said like they are doing disease incidence, right? So
Yeah.
they are basically counting, right? So they are looking at the incidence, right? So basically categorical data. So you'll have to use different, yeah, I would use some sort of like a generalized linear model type analysis. So I'll have to think about and look at the size of the, I mean, you said like a 42 treatment combinations. I mean, that sounds huge, but within that framework, then how to do the multiple comparisons that you have. I guess even like descriptive method, or I guess the summary statistics will give you a lot of idea. Maybe you'll have to use some graphical techniques because you have so many treatments. Yeah, you'll have to use some sort of like a incidence rate across different groups and use some sort of like a graphical technique in terms of presentation. So in terms of tests, then I'll have to think about it because you have so many treatments, but because it's a factorial experiment, then obviously I would look for some sort of interaction. But then can you really do I mean, you'll have to create so many dummy variables in a way. If you were to do some sort of generalized linear modeling for this, because you have so many treatments.
All right, thank you, Dr. Budhathoki. Thank you for your excellent presentation and excellent interaction with our mini grant recipients and advisor and other members. And as you can see, his Dr. Budhathoki's email in the slide that he has shared. You can, if you have any question, please feel free to email him. He would be, very glad to answer your questions if you have any. With this, let us close today's presentation. Once again, thank you, Dr. Budhathoki, for your excellent presentation. Vice President Dr. Wagle, I would like to invite you for your brief remark. Also thank everyone, including Dr. Budhathoki, and adjourn today's meeting.
Thank you, Dr. Gamre, and thank you so much, Dr. Budhathoki, for very nice presentation. अनि हाम्रो नेपालबाट भाइ बहिनीहरूले अब डिपेन्डिङ अन अब त्यो कुन सेमेस्टरमा हुनुहुन्छ त्यसले गर्दा कति बुझ्नुभयो कति बुझ्नु भएन हैन तर इट्स ओभरअल आई वाज भेरी सिम्पल and I mean you try to present in a very simple and clear way so that way they can get some understanding of these methods and all that. So and then if you have any questions, definitely there are questions because you are just learning and I mean even for us we don't know because we learn by practice, right? So even after PhD and all that, whatever we are doing, we understand that, we experience that and all that. But if there's something new, then we have to kind of scratch our head because we don't know what to do and what to look at. So that's how. If you have any questions, probably your advisors also know because they might be doing the similar works, right? So that's why you come up with that type of design. So they might be helpful because they might be doing the similar work. If not, then we can definitely help you to explore further. And then like Dr. Budhathoki said, he had posted his email also, so he will also try to help you that way. Or you can also contact NAPA also, and we can try to find some appropriate person and link with you for that to address your questions. With that, यो लर्निङ प्रोसेस हो त्यो हुनाले जति काम गर्दै गइन्छ त्यति सिक्दै गइन्छ त्यो हुनाले त्यसमा चाहिँ मलाई आएन भन्ने कुरा हुँदैन यु विल लर्न बाइ डुइङ त्यसैले फिल फ्री टु आस्क हैन आफ्नो एडभाइजर साथीभाइहरू प्लस सिनियर्स with that thank you everyone for joining this program today and hope it was, I mean, you learned a lot, and you will be learning a lot again in the future programs. With that, हाम्रो नेपालमा हुनेहरूलाई have a good day भन्दै अब हाम्रो US क्यानाडामा हुने जुन नर्थ अमेरिकामा हुनेहरूलाई चाहिँ good night and have a good weekend. Thank you.
Bye.
Yeah. हाम्रो रिसर्च मिनी ग्रान्ट भाइ बहिनीहरू, हामी अब तपाईँहरूलाई कोरोनाले गर्दा रिसर्चको कामहरू कतिपयले चाहिँ सुरु गर्न पाउनु भएको छैन, अथवा कतिपयलाई गाह्रो च्यालेन्जिङ भइरहेछ। तर बिस्तारै बिस्तारै हामी मिड टर्म रिभ्यूको पनि प्रिपरेसन गर्नुपर्ने हुन्छ, के अरे मिड टर्म रिभ्यू गर्नुपर्ने हुन्छ। त्यसकारण गेट रेडी फर द्याट है। हामीले अब 1-2 महिना कहिले हुन्छ त्यसको लागि बिस्तारै प्रिपेयर पनि गर्दै जानु होला। हामी जुन जुन अवस्थामा छौँ, जे छौँ, हामीले चाहिँ आफ्नो प्रोग्रेसहरू चाहिँ त्यो सेयर गर्नुपर्ने हुन्छ। हजुर,
यसको साथसाथै म के पनि भन्न चाहन्छु भने अगाडि भाइ बहिनीहरूले लोकल एडभाइजरसँग त यु आर इन क्लोज कन्ट्याक्ट बिकज यु आर देयर लोकल्ली, तर नापा एडभाइजरसँग पनि अलिकति फ्रिक्वेन्टली interaction बढाएर perhaps you can get little more both learning as well as getting your moving your project forward. So I highly encourage you to engage, and we all are busy in different things. र भाइ बहिनीहरूले नै चाहिँ घचघच्याएर oh, I need this, I need that भनेर गर्न म इन्करेज गर्छु, ताकि एडभाइजर विल बी quote unquote forced to help you. I just want to encourage that. Thank you.
अर्को कुरा यहाँ अब भाइ बहिनी लगायत अरू हाम्रो एडभाइजर साथीहरू पनि हामीले अब नापाले आउँदो 3rd बाइएनल कन्फरेन्स मे 27बाट 30 मा स्केड्युल गरिसकेका छ। त्यसमा पनि आफ्नो प्रेजेन्टेसनहरू अथवा सबमिसनहरू गर्नको लागि तुरुन्तै कल हामी केही समयमै निकाल्छौँ हैन मेक्सा? अनि त्यसको लागि पनि यो चाहिँ यो पूर्व जानकारी।
डेफिनेटली, एब्सोल्युटली, एब्सोल्युटली। यसैमा अब एड गर्दा गर्दै मैले के पनि भन्न चाहे भने लट अफ यु आर अलरेडी फ्यामिलियर कि हाम्रो आफ्नै जर्नल अघि डाक्टर बुढाथोकी स्पिकरले पनि भन्नुभएको थियो, नापाको आफ्नै जर्नल छ, त्यसको लागि पनि you can begin reviewing that journal's